And we were finishing up speaking about the osmotic pressure, and specifically the way that osmotic pressure can be used to drive water movement, right? So we said that the example here was across the gastrointestinal tract. This is actually the mechanism by which we absorb water. So anytime you drink water or fluids that are, containing water, this is the mechanism by which they are absorbed. So the epithelial cells of your gut will first pump solutes into the interstitial area, which creates a solute/water gradient, and then water will simply move by osmosis down that gradient, right, from a place of high concentration, because there are fewer solutes in the lumen, over into the interstitial fluid to a place of high solute concentration or lower water concentration. So that is the mechanism by which we absorb water, using that knowledge that osmotic pressure creates that pull, and then water will follow. And the rule that we usually say, especially when you talk about the water movement in the kidneys as well, is that water follows salt. So the body knows this, and it intentionally creates solute gradients so that water can follow those gradients as well. Okay. Which is another reason that if you have a very high sodium diet, very high salt diet, you can become dehydrated, because you're not allowing those solute gradients to act accordingly, and then you're not allowed to absorb as much water as the body needs. If we think about another way that this is applicable clinically, we can look at the disease cystic fibrosis. So cystic fibrosis is where we have problems with the export of chloride. So there's a blockage of these chloride channels. And chloride is one of the solutes that we can pump into the lumen here, and that will help us move water into the lumen, right? So again, that solute gradient created first, and then water simply follows. So the normal way that this behaves is that chloride and sodium are pumped out. That creates NaCl in the lumen, and then water will follow that gradient. Now, when we have a defective chloride channel, we are not able to effectively pump chloride or sodium into the lumen. And so the mucus is there, but it's very thick. There's not enough water to dilute that mucus necessarily. And so that's a very thick, viscous mucus, and that can have a lot of clinical manifestations. So these patients usually have frequent recurring upper respiratory tract infections. They may also get lower respiratory infections. They can also have reproductive consequences like infertility, because the same mechanism that allows for, let's say, sperm to be traveling along the reproductive tract, or even the ovum to travel along the reproductive tract, the same mechanism is defective, and so that mucus becomes very thick and almost impenetrable, and not easily able to travel along those reproductive and respiratory tracts. So these patients typically present with recurrent infections, infertility as some of the main symptoms before they're diagnosed. And this is a genetic disorder, so usually you'll have a history in your family of someone who presented similarly, and then they'll do genetic testing and find out you have that defective gene. And so I believe there are enzymes and certain therapeutics that can be used to correct this nowadays. Okay, so that's one of the ways that knowledge of this gradient can help us understand the normal physiology as well as the abnormal, when things go wrong and we have diseases. All right, last thing we're going to discuss here is one of the common mechanisms of movement. So transcytosis. We've talked about facilitated diffusion, we've talked about simple diffusion going right through the membrane, we've talked about channels, carriers, etc. One of the last mechanisms of transport, and we'll speak about some examples of this, especially in like the endocrine system, or even when we talk about neurons, and that is transcytosis. That is the movement of molecules across the cell membrane and even through the cell via a vesicle. So these vesicles are created via endocytosis, which is the proximal part or proximal process that allows molecules to be uptaken into the cell across the apical surface. And that is basically pinching off a piece of the membrane. So here's our molecule. We want to get it moved into the cell and eventually into the blood. So we pinch off a piece of the membrane, right, the phospholipid bilayer that's at the apical surface, which creates this vesicle where this molecule can travel inside the vesicle, across the cell, and then the opposite end, the basolateral surface, it's going to be exocytosed. So it's going to be dumped outside the cell by the opposite process, exocytosis, and then the vesicle membrane will fuse with the cell membrane once again, and then that molecule will be dumped onto the basolateral surface and then eventually into the blood. So endocytosis and exocytosis together allow for transcytosis of molecules, and this is where they're incorporated in this vesicle. And again, this is going to be very important when we talk about the way that neurons communicate. So we will speak about how neurotransmitters get packaged into these vesicles. They get dumped via exocytosis. They travel across to the adjacent cell membrane. And this is how neurons speak to each other, right? By creating these neurotransmitter vesicles that can be relayed across the synaptic junction. All right, so this will come back again as an example.